Chapter 18, Section 1, The Metabolic Fates of Amino Groups. Okay, we have discussed and touched on in previous chapters how the amino acids are going to be broken down and via catabolism, using different metabolic processes, and how they can be fed into different metabolic processes. You take, here, glutamine. Glutamine can be, modified, to become glutamate. Glutamate to alpha-ketoglutarate. Alpha-ketoglutarate, part of the citric acid cycle, will then be, processed, carbons pulled off, excited electrons pulled off, then it's going to be repackaged back to oxaloacetate so that more citric acid cycle can take. Or the alpha-ketoglutarate can be modified to become pyruvate. Pyruvate, which can then be turned back into glucose 6-phosphate using the gluconeogenesis. The thing is, though, these amino groups, you're not going to find those on any of the intermediates of glycolysis, gluconeogenesis, nor fatty acid synthesis. So while these carbon strands are going to be great, these amino groups, NH2, NH3, we're going to have to get rid of those. Our cells are going to have to get rid of those in some form or fashion. Now, great, without a doubt, we have enzymes that can remove, this amino group here, turn this into a carboxylic acid functional group. But the thing is, what do you do with that nitrogen-containing compound? Well, easiest thing is turn it into ammonia. Okay. The problem is, most aquatic animals can get rid of the ammonia very easily. They're in an aquatic environment. Okay, it will diffuse out very quickly, or they can pass it into their bloodstream, and out the cloaca. Easy peasy. For us terrestrial animals, since we're not walking around constantly urinating on ourselves, at least I hope you're not. If you do, you really should see a doctor about that. We're going to take this ammonia and we're going to repackage it, process it, and we're going to form urea. Okay, urea, hence urine. This is how our kidneys will remove the nitrogen-containing compounds. Ammonia from the cell becomes urea, which, kidneys take out, put into the bladder, out with excess salts, sugars, and all the other stuff in your urine. And this bad boy you see here just above my head, this is uric acid. Okay, because it comes out, birds don't urinate. Birds, many reptiles, do not urinate. What they do is they concentrate the ammonia into this uric acid that comes out as part of the feces, the bird droppings. If you're anybody who's ever had a car parked out on the street and gets covered in bird droppings and then wonder why their paint job got all splotchy, right there, uric acid. Uric acid ate away at the wax, ate away at the surface layers of the paint. The more you know. So, as I said, most aquatic animals, very simplified. Since we need to store it for a bit, we need to take it out of our fluids, store it a bit, and we do it as, urea, the uric acid. Think birds, animals. Anybody who's had a pet bird, had a pet animal, you'll see they leave those. It's not always white droppings, but it's this clumpish, gelatinous goo of droppings. There you go. Well, that's going to be very high in nitrogen. Makes sense that if you're trying to get nitrogen out of biological compounds, the ones that are going to have the most nitrogen that you are definitely not going to be recycling is going to be the amino acids. So what we found as we have studied cell metabolism, cell biology, that alpha ketoglutarate seems to be one of the intermediates that's the easiest to replicate. Any of the amino acids that can become glutamate or glutamine with, a cut, a paste, a twist, a tweak, maybe moving a hydroxyl group, something like that. Well, the glutamate, the glutamine becomes alpha ketoglutarate, as we saw in the last slide. Or you can go on alanine to pyruvate, aspartate to oxaloacetate. Here's our key citric acid cycle intermediates. Pyruvate feeds in. Oxaloacetate takes the acetyl-CoA, which pyruvate will be converted into. And then you get citrate, isocitrate, and so on, alpha ketoglutarate, which, a couple of more steps of removing carbon, removing a couple of more electrons, pull off some energy and phosphorylate GTP, and then you're on your way to rebuilding oxaloacetate. We see that when it comes to the proteins, the polypeptides, that the degradation, the beginning of the breakdown process, actually happens in the gastrointestinal tract. Before we're anywhere near protein or the cells in your body, the work starts in the stomach. Gastrin kicks off the party. Gastrin's a hormone. It's released when cells lining your stomach are triggered to the presence of high levels of protein. Okay, so that means you're eating protein-rich diet. Okay, stimulates secretion of HCl and pepsinogen. Okay, hopefully you remember from last semester, if you took biochem last semester with me, or from your cell biology, that many of the enzymes in your body are not translated into a working protein. They're translated into a protein that sits and waits. Waits until it gets cleaved. A part of it gets cut off. And once that part gets cut off, it is now an active enzyme. Those are known as zymogens. Those are enzymes to be. Having that little bit left on there means that they're not quite the right shape yet. That last little bit hasn't been cut off yet, keeping it from functioning, keeping it from working. So pepsinogen is a zymogen of the enzyme pepsin. Autolytically cleaves itself. Autolytically means it self-cuts. Self-lytic cuts. It does so because of low pH. Gastrin causes increase of HCl concentration in the stomach, which lowers the pH. Lowering of the pH means that the pepsinogen that's now also been upregulated, meaning more will be made, it's going to cleave itself and become pepsin. All of this is happening here, as I said, in the stomach. Parietal, here you have the columnar epithelial cells. I believe it's the columnar epithelial cells. Sorry, A&P was many, many decades ago. You have the columnar epithelial cells lining the stomach. Certain cells that make up this epithelial lining, parietal cell, chief cells, gastric cells. amongst many other. They're not showing here the goblet cells that make the mucus that protects the lining. Starts off with the mucosal cells here, secreting gastrin. Gastrin then triggers the chief cells and the parietal cells to start making and start pumping out, excreting HCl and pepsinogen. Pepsinogen autolytically cleaves itself into pepsin. Pepsin then goes and starts cutting long polypeptide chains, making a much smaller. Long, short, what's the numbers? Don't know. I'm sure somebody has sat down and studied the numbers, but honestly, I can't remember. Because think about it, you have some of these polypeptides that are making up proteins that are going to be hundreds of amino acids in length. There's no way that those hundreds of amino acids are going to be taken up by any of the cells. They're just too big. So go through and start cutting, cut. Make them much shorter, a dozen or so. I'm just using that in numbers. Don't quote me on those. Amino acids in length. Stomach pH drops because of increased HCl production. HCl causes the pepsinogen to autolytically cleave to pepsin. Pepsin goes through along with the slurry of the food and is cutting up polypeptides. So you get these zymogens that are becoming active proteases going through, cutting as it's going from the stomach through the duodenum and everything into the small intestine. All right. In the presence here, as this lower pH comes in through here, cells here, or I'm sorry, this lowered pH, meaning there's a bolus of food going in and a bolus of food's now moving through, will trigger the pancreas to start producing and releasing the hormone known as secretin. Secretin stimulates the release of bicarbonate into the small intestine. Low pH in the stomach, good. Low pH in the small intestine, bad. Why do you not want the low pH in your small intestine? Well, as I mentioned in the last slide, there are goblet cells here, lots of goblet cells lining the stomach, producing a mucus that's forming a layer of protection between the stomach contents and the mucosal lining. Here, not as many goblet cells. Yes, there's still going to be some mucus here acting as a lubricant, keep things moving, but not as much. The cells here are going to be in the different than the ones here. They're not going to like that low battery acid level pH. Having the low pH here in the stomach means you're breaking things down, not so much absorbing. Low pH here will interfere with the absorption of many compounds, many chemicals and things that are in that bolus of food passing through. So you want to get this not neutral, but at least you want to get that pH up. Can't be around the 2 that it is in the stomach. So as you see here, low pH, pH 7, because of the work of the pancreas causing bicarbonate to be released, start bringing that pH closer to near neutral. It turns out the zymogens, there's quite a few of them that are coming into play here when it comes to our protein catabolism and amino acid utilization. When we look here, cholecystokinin, I mispronounce this one every time. Cholecystokinin. Wow, cholecystokinin. I really hate some of these words. Cholecystokinin is hormone secreted in the blood. Okay. When the cells lining the duodenum, remember the stomach, jejunum, duodenum, small intestine, well, when the cells there lining the duodenum start getting the receptors ping, ping, ping to high levels of peptides, high levels of amino acids, proteins, going through and as a bolus of whatever is in the stomach. So this hormone gets secreted in the blood and then stimulates again the pancreas. Pancreas that, remember, did the release of bicarbonate. Also is looking at releasing of many other zymogens. Trypsinogen, chymotrypsinogen, procarboxypeptidases. These will then, also autolytically cleave themselves, trypsin, chymotrypsin, carboxypeptidase A and B, which will then go through and start further breaking down the shortened polypeptides. We also see that even though many of these can autolytically cleave, they do have some helpers. Enteropeptidase will go through and convert trypsinogen to trypsin. The trypsin will then go and start also cutting more, making more trypsinogen, cutting trypsinogen, the chymotrypsin, and the procarboxylases and proelastases. So it's almost a domino effect. Yes, many of these can do autolytic cleavage. It takes time. Or you can cut one, and then that one goes and helps the others, trypsinogen, chymotrypsinogen, carboxypeptidases, proelastases, to further quicken the pace of cleavage. So now instead of full-length proteins or, a mature protein now releasing its multiple polypeptides because the quaternary structure, all right, quaternary structure is multiple polypeptides, but those polypeptides are still going to be 100, 200, 300 amino acids in length, which is still too big. So then you get the trypsinogen and the chymotrypsin, which are going to come through and start cutting, along with all the others that we just talked about. And they're going to cut and release. Go through and cut every peptide bond, boom, 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 of the polypeptides, giving free amino acids. The free amino acids that the cells lining the mucosa can then take up individually. They'll be passed through these cells through the basal lamina into the underlying tissues, which are going to be highly vascularized to take away those amino acids for further processing elsewhere. As they are being taken elsewhere in the body, first trip they're going to go through, all of them, is through the liver to make sure there's nothing along with the protein, those amino acids, that could be damaging to the rest of the body. Because remember, the liver's job is to take the brunt of the damage of everything you've eaten and then absorbed from that food you've just ate. One of the things that we see that people run into is an issue known as acute pancreatitis. So the cells lining the duodenum and whatnot are still sending out the signals, and the pancreas is still releasing all of those hormones and all of those zymogens. Just nothing's getting out. Or if it's getting out, the zymogens, so let me step that back. So either there's a clog in the system, so things aren't getting out, so the pancreas is increasing in size, and things start triggering, the zymogens start triggering and starts damaging self. Or even without food, the proteins going into the stomach, passing into the duodenum. The pancreas, the cells of the pancreas are prematurely converting the zymogens that are supposed to be stored away. Make a lot now, pack them up, store them so that they can be released all at once in high concentrations, so that they can then cleave and become active proteins, a lot of them all at once. Well, it turns out that instead of storing them, they're just activating them, and things are being released into the pancreas itself as active or into the small intestine when there's no food there. And you're doing some intestinal damage that way. So acute pancreatitis. A lot of this, anything ends in itis, means inflammation of. So this is acute, fast onset, short time frame inflammation / infection of the pancreas.